Welcome to a deep dive into the complexities of canine neurological health. Today, we explore a groundbreaking study focusing on necrotizing meningoencephalitis, or NME, a devastating brain disease affecting our beloved dogs, particularly young, small breeds. This research, conducted at the University of Pennsylvania, aims to unravel the mysteries of NME and pave the way for improved diagnostics and treatments. NME is a type of meningoencephalitis of unknown origin, or MUO, a category that includes other conditions such as Granulomatous Meningoencephalomyelitis or GME and Necrotizing Leukoencephalitis, or NLE. These diseases are frustrating for both veterinarians and pet owners, because diagnosis can be challenging and is often only definitively made post-mortem through histopathological examination. Current treatments for MUO usually involve immunosuppressive drugs, including corticosteroids, sometimes combined with chemotherapy, and supportive care. The study we are about to discuss dives deep into the unique pathological features of NME compared to GME to try to improve diagnostic accuracy and therapeutic approaches. The researchers began by outlining their primary objective: to identify distinct disease mechanisms within each MUO subtype. They did this by studying the degree of immune cell infiltration, astrogliosis, demyelination, and microglial activation. This was achieved by comparing NME with GME, a closely related MUO subtype. Study design: the study's design involved a meticulous examination of brain tissue samples from dogs diagnosed with NME and GME, using the archives of the Penn Vet Diagnostic Labs. The researchers included 11 NME cases, 11 GME cases, and 5 neurologically normal dogs as controls. These cases had been confirmed post-mortem by veterinary pathologists, based on clinical signs, signalment, and histologic findings. Formalin-fixed and paraffin-embedded or FFPE tissue blocks were used, which were sectioned into thin slices, mounted on slides, and stained. The sections were then stained with hematoxylin and eosin or HNE for general visualization, and then with specific antibodies to examine immune cells, myelin, and glial cells. These antibodies targeted CD3 for T cells, CD79B for B cells, IBA1 for macrophages and microglia, myelin oligodendrocyte glycoprotein or MOG for myelin, glial fibrillary acidic protein or GFAP for astrocytes, and aquaporin 4 or AQP4, another astrocytic protein. The immunostain slides were scanned with a digital scanner to allow for detailed analysis of the stained areas. Quantitative analysis: to quantify these stains, the researchers used a positive pixel count algorithm on the digital slides, focusing on specific annotated regions to assess demyelination, astrocyte activation, and AQP4 expression. The extent of inflammatory infiltrates in the leptomeninges and brain parenchyma were scored using a tiered system. The leptomeningeal infiltrates were scored as absent, mild, moderate, or marked. The parenchymal inflammation was similarly scored, based on the presence of perivascular cuffing. Glial nodules, which are clusters of immune cells, were also scored based on the number of nodules, and number of cells within each nodule. Finally, the collected data were statistically analyzed to identify any significant differences between the control, GME and NME cases. Results: the results of this investigation revealed some key differences between NME and GME. First, the team found that NME is characterized by mild immune cell infiltration in the leptomeninges, in contrast to the prominent, B-cell-rich aggregates seen in GME. Leptomeninges are the membranes surrounding the brain and spinal cord. This observation is demonstrated by Figure 1, where in panel A and B, HNE stained slides reveal inflammatory infiltrates, and C, the quantified data showed that GME has significantly higher levels of total inflammation in the leptomeninges compared to NME. Panels D and E show the B-cell infiltrates stained with anti-CD79B, and F, the quantified data demonstrates significantly higher B-cell infiltration in GME compared to NME. In contrast, T-cell infiltration, as shown in panel G and H stained with anti-CD3, were similar in both groups, with the quantified data in panel I showing slightly higher levels of T cells in GME than NME. Moving to the brain tissue itself, or the neuroparenchyma, both NME and GME showed comparable lymphocyte infiltration. However, Figure 2 shows that although both conditions had inflammatory cells in the neuroparenchyma in A and B, the quantified data in panel C showed that total neuroparenchymal inflammation is similar between the two groups. The data for the B cells shown in D and E, quantified in F, and the T cells shown in G and H, quantified in I, were similar between the two groups. However, demyelination, or loss of myelin, was significantly more pronounced in NME, particularly in the subcortical white matter. Myelin is the protective layer around nerve fibers, and its loss disrupts the normal transmission of signals in the nervous system. This is highlighted in Figure 3, where the images in panels A and B show control and NME tissue stained with anti-MOG, a marker for myelin. The quantified data in panels C through F demonstrates that the demyelination is much greater in NME, specifically in the subpial gray matter and subcortical white matter, compared to GME. A significant finding of this study was that areas of the brain affected by NME displayed a reduction in astrogliosis, that is, less activation of astrocytes. Astrocytes are star-shaped cells in the brain that support other cells, and normally become activated during inflammation. In addition, there was a marked decrease in the expression of the water channel protein aquaporin-4 or AQP4 in NME, a reduction not seen in GME. The expression of AQP4 correlated with the extent of microglial and macrophage activation. This is demonstrated in Figure 4, where panels A and B show control and NME tissue stained with anti-GFAP, a marker for astrocytes. The quantified data in panels C through F demonstrates that GFAP expression is significantly lower in NME compared to controls, throughout the cortex and subcortical white matter, and trends lower compared to GME. Furthermore, Figure 5, where panels A and B show control and NME tissue stained with anti-AQP4, and C through F showing the quantified data, demonstrated that AQP4 expression was reduced in NME, specifically at the subpial and leucocortical areas. The study also looked at correlations between the various markers. In the cortex, there was no correlation between loss of myelin and loss of GFAP staining, nor between GFAP and AQP4 or myelin and AQP4. However, in the subcortical white matter, a loss of GFAP expression correlated significantly with a decrease in myelin, and the same was seen for the correlation between AQP4 GFAP and AQP4 MOG, which suggests that damage to both astrocytes and oligodendrocytes may be related to a decrease in AQP4 expression. This data is shown in Figure 6. Panel A shows the correlation between MOG and GFAP in the cortex, B shows the correlation between GFAP and AQP4 in the cortex, and C shows the correlation between MOG and AQP4 in the cortex. Panels D through F show the same correlations in the subcortical white matter. Finally, the team examined microglial activation, using the IBA1 antibody. They found that while there was no association between the number of glial nodules and loss of myelin in the cortex, in the subcortical white matter, expression of myelin decreased with increasing number of glial nodules. In addition, they found that expression of AQP4 was negatively correlated with glial nodules in the cortex and trended towards the same correlation in the subcortical white matter. The data for this is shown in Figure 7, where Panel A shows the correlation of glial nodules with MOG expression in the cortex. Panel B shows the correlation of glial nodules with AQP4 expression in the cortex, and Panels C and D show the same correlations in the subcortical white matter. Finally, they examined the correlation between lymphocyte infiltration and AQP4, finding that increased inflammation was correlated with decreased AQP4 expression in the cortex but not in the subcortical white matter. This data is shown in Figure 8, where panels A and B show the correlation between AQP4 and B and T-cell infiltration in the cortex, and C and D shows the same correlation in the subcortical white matter. Discussion these findings led the researchers to some important conclusions about the unique features of NME compared to GME. Although the cause of MUO remains largely unknown, the most abundant body of evidence suggests NME to be either a primary autoimmune disease or a multifactorial immune-mediated disease. There are some genetic risk factors that predispose dogs to develop NME. Also, there is evidence of an autoantibody against GFAP found in cerebrospinal fluid and serum. Whether these antibodies are directly causing the lesions or are produced secondarily is still unknown. The study confirmed that while the immune cell infiltrate in the brain is similar in both GME and NME, the infiltration in the leptomeninges is distinctly different. GME is characterized by the presence of B cells, and NME has far less infiltration and with no predominant lymphocyte type. The presence of B cells is important, as it suggests a chronic process and a production of antibodies which could lead to a cyclical immune process. It was also confirmed, similar to the findings in GME, that demyelination is a key feature in NME. The researchers examined areas adjacent to the necrotic lesions in NME and confirmed that demyelination was extensive, particularly in the subpile gray matter and subcortical white matter. A striking finding was the reduction of astrogliosis in NME, which is typically increased in other neuroinflammatory conditions. The researchers suggest that in NME, astrocytes are the likely target of the immune response, and this could be why there is a reduction in GFAP and AQP4 expression. Indeed, the loss of AQP4 correlated with the presence of glial nodules, suggesting that astrocytes shift away from their normal homeostatic functions to a pro-inflammatory role. Finally, the reduction of AQP4 also correlated with increased B and T cell infiltration in the cortex, similar to findings in human neuroinflammatory disorders. This suggests that astrocyte damage might initiate a pro-inflammatory state leading to activation of microglia and perpetuation of inflammation. Clinical implications. This research suggests that NME is more than just an inflammatory disorder of the brain, but also involves significant astrocyte dysfunction. The unique finding of reduced AQP4 expression is a potential marker that could help differentiate NME from other MUO disorders. This opens doors for the development of targeted therapies to address not only the inflammation, but also the specific role of astrocytes in NME. The researchers suggest that premortem studies looking at the specificity of IgG in cerebrospinal fluid and serum may lead to a diagnostic biomarker that could help in early diagnosis and development of targeted therapies. The research also highlights similarities with autoimmune GFAP astrocytopathy in humans, which is characterized by B and T cell infiltration and loss of GFAP and AQP4, suggesting a similar disease process in dogs. This detailed examination of NME and GME in dogs provides new and essential information to help diagnose and treat these heartbreaking conditions. By focusing on the unique features of NME, particularly the astrocyte dysfunction and AQP4 depletion, scientists are hopeful to improve the lives of dogs affected by this disease. From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.